Our next speaker is Lon Cardon, who is the Senior Vice President of Alternative Discovery and Development and Head of Target Sciences at GlaxoSmithKline. Lon is also an advisor to the Precision Medicine Initiative, so he wears two hats in this conversation.
Well, thanks very much. It's a pleasure to be here as always. I've been here a few times before. So Nadim issued an interesting charge at the beginning to think of one thing that would disrupt the path from translating genetics into new medicines in 3 to 5 years. I found that really refreshing because the null hypothesis is that it will work. And what do we find that takes it off that path? Now that's a big change from where we were in this field not very long ago at all, which was what is it in genetics, or is genetics going to deliver? And so I wanted to start by raising a few of these points to set up something that I want to talk about, which are a few collaborative efforts that we've got going to address some specific problems. Let me set the problems up. The way I look at it is to Kari's point last time, last session, it's unquestionable that genetics has just had a transformation over the past decade. 15 years ago, we used to talk about whether there were 7 versus 10 genes for common complex diseases that we knew about. And now we've got thousands that we can ascribe. No question about that. Has the translation met that same success? I think that's a much different situation. And I think we all in this room expect and hope that it is on the path to do so, and that we just haven't seen the fruits of those labors yet. But we have heard a number of speakers today already say there are a few examples, but not a lot in translation. And I will say, I need to say right off the bat here, for some areas, that is not the case, and translation's been fantastic. Look at oncology, at least targeted oncology therapeutics. Look at rare diseases where cell and gene therapy are opening them up. Some of the side effects, the significant big side effects that come through, if they're large and they make it through on things like cancer drugs, they're detectable and people find them. So those are great successes, and genomics has really won there. So I'm referring to the rest of it. So what is the translation opportunity there? And in fact, I just thought I would, if I can make that work, throw in a paper came out last week describing pharmacogenetics, just as an example of where we are today. And this shows in the, I believe, the blue blobs, the genes that have a big impact for efficacy in pharmacogenetics. The red ones are safety. The point there is not many blue ones, and a lot of safety. Okay, so that's just one area, but we haven't yet translated in these, and yet we've got lots of success in the disease gene finding arena, a great deal of success in this. So where is genetics going to have its promise, and what are the first areas of translation that the impact is going to be beyond oncology and rare diseases and an adverse event? Now, I think it is around targets. I think it's around target validation. We've danced around this in almost all the talks thus far. And I'll exemplify it by this. I think this was a really important paper, not least of which because I was co-author, but because I think it shows something that's really important. Matt Nelson and others last year, a couple of years ago, they did this. Last year put out this paper that said, all things being equal, from the very first stage you start in drug discovery, the choice of the target that you make, all things being equal, if you take those that have some form of genetic evidence, even dirty GWAS type of data, evidence where we don't know the gene localization particularly well or the necessary mutations, your chances of getting to a successful drug double, 2x-fold, just by taking those that are genetically associated or validated in some way, crudely, double your chances of success. That doesn't sound much probably to the epidemiologists in the room, but it's the biggest predictor we've got, actually. The only other thing that predicts drug success better than that is if that gene had been drugged before, right? So this is the best information we have, actually. So why wouldn't you use this if you were going into a drug discovery campaign? And in fact, you might think I blocked out this on the bottom, sorry. All pharmaceutical companies do this, but actually on average, if you look at the data that are in clinical trials today, these are everything in there, but the blue ones, the little blue bits on the bottom, about 15% of the targets that are in trials today have that genetic information. There's an interesting thing we won't talk about, that the red stuff in there are those same targets that are associated with something that's not the indication they're being pursued for. So there's genetic evidence, but it's not the one, at least it's not the specific trait. But it means most of the things that are being conducted today do not have that, they're not enjoying that twofold increase in probability of success, which is pretty surprising. Now, there are those in this audience I recognize that do good pharmacoeconomic modeling. if you take that same premise of twofold increase in success, Nadeem started the day by saying it costs a lot of money and it takes a lot of risk. If we went from 15% of the targets to just half of them as an industry or as a community, then you drop about 15% of the overall cost to every successful drug. If you think drugs cost a couple of $1 billion to get there, for every single success, you're going to knock 15% off that, just by changing the first stage of drug discovery. So you can make economic arguments, you can make scientific arguments, you can make common sense arguments that is an area where this should all work. Okay? So that's the background here. You might think then that this is a big spiel and propaganda argument for GWAS and for pursuing everything in that. But where we left again, the previous session, was that it's not that easy. Not all genes, this slide really should say, not all genes are good drug targets rather than targets. It's really hard, and still we fall into the trap as a community. Read the next paper, and you'll have the next GWAS paper or gene discovery paper. We'll say at the bottom, this is a good target for disease X. It's not necessarily a good target for disease X. It's a good predictor of disease X, but there's a whole bunch more to it in turning that predictor into a valid drug target. And it may well be the case that we want to pursue these for 25 years. Absolutely true. But they don't all just pop out. And in fact, the examples of those that are coming out of the GWAS screens that we see today, a lot of them are really, really hard to drug. And the people in the chemistry world will look at those in 2 seconds and say, at least small molecule world, will say, you know, that's going to be a 10-year med chem program if we really want to take that on. And they'll know because they've been there and they've seen that. And so we can't say that every time we find a new association for a disease, that's a drug target. At least it's not a good one, not necessarily a good one. A lot further to go. So these sort of simple observations, if I can get this to work. I'll come back to that actually. These simple observations a few years ago led us to try and think about how we might improve the situation and actually capitalize on the data that we have in front of us. And the situation was, clearly, for targets that we might take into the clinic or that we pursue, genetics, it's its time. It's come through its hype cycle. It's back up the other end. There's enough there that there's enough to choose from that we think we can choose better targets. But it's not getting easier. Actually, it's getting harder. The specialization's getting harder. We have expression coming in. The slide I just skipped was about gene regulation and localization, and not all of these are protein coding changes, and they're pointing to things that are outside regions that we would have even anticipated. It's getting really hard, so it's hard to have all that specialist expertise in one place. We certainly don't have it in big pharma, not all of it to get there. We have drug discovery expertise, maybe some biological expertise, but the genetics and the genomics and the technology and the sequencing, they're all going in a more specialist direction. So we were recognizing that. And then we made a key point that is a claim that GSK has very strongly. Some other pharma agree with this, others don't. That the targets themselves, so not just the genes, but the things we choose to work on and the annotation thereof, what they're doing, how they interact, the pathways we just heard about, those are pretty competitive and we'll share them. So when I first went to GSK, I don't know, 7 years ago or something, the first thing I did was dive in, do some sequencing, and publish every single target we were working on. Just made it available. It's all yours. There's no secrecy there. It's not the target itself that our argument is that we would compete on. We compete on the drug discovery side of things, the chemistry and the clinical trials and the creativity and linking that target to interesting phenotypes. But the targets themselves are better pursued by all of these areas of expertise. It's a very key and fundamental point, and it's not, again, one that may well come up by others because it's not shared by all. But if you take that premise, it really opens things up, because then you can imagine a world where you have expertise in drug discovery operating next to expertise in the most fundamental genomics operating next to the EHR experts of the world, all asking different questions of the data, which is where I was really trying to go with my own question. How do we get access to all of these things so everyone from a different perspective can get into the business and understand how best to choose the things to work on and take them forward? So with those simple principles in mind, we set out to try and create an environment where that could happen. And we created this center called the Center for Therapeutic Target Validation, which was really meant simply to be an environment where, as I just said, you put drug discovery people next to the specialists in the field, all sharing all stories, let all flowers bloom, and the results of those, because they're pre-competitive, can go to whomever chooses to pursue them, the academics, the pharma, biotechs, whoever's around. That's what we were trying to create. And so we put this together in Hingston in the UK, because in that particular site we have really good they have really good expertise with the European Bioinformatics Institute, EMBL, on large data sets, bring all the public data sets that we can together. They've got the Sanger with all this genome expertise from decades of doing the sequencing. And then you bring pharma, and you literally put them on one site and let the projects bloom from there. That was the idea. And that's what we've been pursuing for now 18 months, to really charge those individuals with whatever projects are appropriate, and we'll all share and we'll all take it in any way we possibly choose to do so. The idea on that was always to be something that would share across pharma, across biotechs, across whoever wants to join in there. We started it just with GSK, with one pharma, because we didn't have the longevity to set up a pharmaceutical consortium, because those things take a very, very long time and a lot of watered down goals as well. So we started it alone, but argued that actually this was for all, and to succeed, we needed to get others in there. We needed others to realize that vision with us. And I'm really happy to say on this one that as of just February, Biogen jumped in. And since Biogen jumped in, the phone is starting to ring, and now others are beginning to join. This, it's a first sign, but it's a really great sign that this may well take off. It demonstrates that this wasn't a GSK-only play. We took the risk of funding it in order to get it off the ground. But now others are joined in on that as well. So that's the Center for Therapeutic Target Validation, and we can talk about specifics and databases and that sort of, but that's the goal, is to really bring together the sophisticated expertise that exists in pockets, but put it all in one place so that everyone can benefit from it. Now, I was introduced, so that's that part. I was introduced to this as wearing two hats on the Precision Medicine Initiative. I'm a new advisor to this initiative, so I'm not a spokesperson. There are others in this room that I will defer to that can speak a lot better to it. I've been around it for a long time. But I do want to make one point clear on this. What this CTTV is meant to do is the earliest stage of drug discovery, so we can better choose what we want to work on. What it does in practice, we can see already, is raise all these interesting questions of what phenotypes might be relevant for this particular variant or this mutation. How can I explore a combination of things in different pathways to bring them all together? It actually cries out for something that is the Precision Medicine Initiative, or the type, or a collection of the type of things that we were talking about just a moment ago. One leads directly to the other. And so I think the Precision Medicine Initiative will follow from these drug discovery target validation activities just about perfectly. And we need to find a way, and where I really want to take this discussion, I'm interested today, is saying how can we make something like this, the PMI, equally accessible to all, so that we can benefit all parties for all indications, for all questions. Thank you.